안녕하세요. 제 이름은 사경하이고요. 저는 지금 삼성서울병원에서 지금 포닥으로 연구를 진행을 하고 있습니다. 그리고 오늘 시작하기 전에 한 가지 이제 양해를 부탁드리는 게, 제가 아직 한국말로 발표하는 게 조금 서툴러서요. 네, 발표만 있는 영어로 오늘 진행하도록 하겠습니다. 네, 감사합니다. So today, the main presentation topic, or the title, is the pharmacogenomic landscape of patient-derived steroids informs precision oncology therapy. So the title is very self-explanatory. In this study, what we did was we compiled a large-scale pharmacology drug response along with genomic characterization of tumors to be able to suggest and identify which therapeutic target is the most optimal for each individual patient. And before I dive really into the actual content, I would like to emphasize I do tend to talk fast while I'm presenting. So if anyone feels I'm going too fast, please don't hesitate to slow me down at any time. And thank you for understanding the situation. So precision medicine involves personalized treatment option for each patient. So before idea of this concept of precision medicine really surfaced, all the cancer patients followed the standard protocol, which was one size fits all. And this was standard treatment option for all the patients. And then, unfortunately, the problem with this kind of treatment option was that not everyone could benefit of this specific treatment, and a lot of people would eventually fall out. So in order to address these challenges and difficulties, they have initiated precision medicine, which molecular characterization of tumors of individual patient can really guide and identify what is the right therapeutic option for each patient. And this concept of precision medicine has really been popularized back in 2016 when former President Barack Obama has started Precision Medicine Initiative. And then the following year, in 2016, when Vice President Joe Biden announced a crucial piece of his Cancer Moonshot Initiative, really overlapped with the Precision Medicine Initiative. And this key component was the opening or starting of Genomic Data Commons data portal, which is commonly known as GDC, is where they have accumulated a massive cancer genome data, which is publicly available to all researchers, clinicians, so that they can use this information to make informed decision for the patient. And also they could use such data for their research of interest. And this accumulated data really has been spearheaded by a lot of different groups, especially led by the world-known the Cancer Genome Atlas and also the International Cancer Genome Consortium. And with their collaborative effort, they were able to produce massive genome data of more than 10,000 patients in 30 different tumor types. And they were able to identify really important recurrent genetic alterations that were crucial in tumor initiation, progression, and survival. And while this was very informative, they were also able to acknowledge how cancer was a very complex disease. It was a disease of the genome with many profound genetic alterations, and a lot of them showed very extensive both inter and intra-tumoral heterogeneity. So intra-tumoral heterogeneity, what it means is that patients can be diagnosed with the same cancer, but they have very different molecular and biological behavior. And because of that, they will behave differently to the same treatment. That is why the one-size-fits-all concept cannot be applied to treat all cancer patients. And thus, the birth of Precision Medicine Initiative has been started. So based on this such knowledge, they have conducted a clinical trial of molecularly targeted therapy based on tumor molecular profiling versus the conventional therapy for clinicians. And then ideally and theoretically, a lot of people were excited and believed that this molecular guided treatment would be far superior in comparison to the treatment at a physician's choice. But unfortunately, there wasn't any significant survival gain advantage in terms of both overall and progression-free survival. And there could be a lot of different reasons and features that need to be accounted for. But the main reason they suspected was that tumor has a lot of different genetic alterations. So it's really difficult to determine what is the right therapeutic target and what kind of target therapy should be given for each patient. So in order to address this kind of effort, they have started to integrate a lot of different data sets. And in recent years, especially led by the Broad Institute and the Wellcome Trust Sanger Institute, they were able to publish a large-scale pharmacogenomic analysis of cancer cell line models. And while they really provide us the reference points how this analytical approach should be done, and while they were very informative, there are a few reasons that cannot be really implemented in the clinical setting. And the two major challenges that really hamper the clinical utility of the current knowledge was that, as I mentioned previously, the first one being cancers harbor multiple molecular aberrations. That's why predicting how patients will react to a certain target therapy based on genomic profile is not enough, because you cannot really represent the whole tumor based on single biopsy of a genomic characterization. And then the second the challenge was that predicting outcome that has been extrapolated from the cell line model it's not sufficient to be able to represent or recapitulate each patient's tumor, because as I mentioned earlier previously, each patient who are diagnosed with the same cancer, they behave very differently. So using cancer cell line models for all these patients may not be such a sufficient way of doing things. So in order to address these difficulties, we have in this study generated and accumulated a very large scale 462 cases of patient sample, in 14 different cancer types that consist of glioblastoma, metastatic brain tumor, breast, lung, stomach, colon, ovary, and so forth. And from there, we were able to extract DNA sequencing information and RNA transcription expression, along with chemical response to 60 different pharmaceutical compounds. And using these gene-drug associations, what we hope to do from this study was that that association can be studied more in depth and can be translated into the clinic, where the clinicians can use this data to be able to predict how patients would react to certain targeted therapy. So this is one of the very important figures that we wanted to convey in this study, was that we first analyzed the both mutation and gene transcriptome expression profile of our patient-derived spheroids, along with TCGA primary tumors, cancer cell line encyclopedia, and GDSC cancer cell line models. And then one of the interesting that we found was that while our patient-derived spheroid models are originated from East Asian Korean patients, all the TCGA, CCLA, GDSC were mostly based on Caucasian population. But our spheroid genomic profile and transcriptome profile showed very consistent results with the TCGA primary tumors. So this gives us a reference point on why should patient-derived spheroids should be used to be able to represent actual patient tumor biology. And another important question was that since we are approaching using targeted therapy, are these molecular targets well retained in the patient-derived spheroids, because we're conducting drug screening of the cells, not the actual tissues. And then we have conducted a genetic similarity analysis between tissue and the matched spheroids, and we found that majority of the cancer driver mutations, including TP53, PTEN, EGFR, PI3 kinase CA, and so forth, they were well retained from tissue to the spheroids. And also, we were able to identify specific gene expressions that were very specific to stromal and immune cells. And after we filtered them out, we were able to show that a lot of the transcript expressions are well preserved from tissue to PDCs. So all these data gives us a good validation and credibility on our systematic approach of using patient-derived spheroids to be able to predict how patient will react in the clinic. So the first approach that we did in our study was, since we have such a large scale of different tumors, we compared each drug response to each individual cancer type. And on the left panel, shown in the y-axis, all individual drugs which has been clustered based on their drug class family has shown a different results on each y-axis, which is a disease type or tumor type. And interestingly, there are some classes of drugs that really acted similarly. So for example, as you can see, EGFR targeted drugs they were very resistant to glioblastoma cells, while they were sensitive to both breast and gastric tumors. So this gives us an idea how to approach each tumor type based on their biological behavior. And then one of the interesting things that we found was that through the topological data analysis shown in the middle panel, we found that BYL719 inhibitor, which is the PI3 kinase inhibitor, was highly sensitive to gastric tumor, but it was resistant to gliomas. And for those who are not quite familiar with the topological data analysis, this topological data analysis, or TDA, is a dimension reduction method. So this is very similar to conventional methods such as PCA or TSNE. What it does is it reduces a lot of the dimensions to be able to represent in a two-dimensional structure. And so what it does is, as you can see, each node or circle here is a cluster or group of tumors that has been clustered based on their gene expression profile. So if a tumor has very similar genetic gene information, they will be clustered into one circle. And each circle or each node is connected by a line or an edge if they share a tumor in common. So this graph 1 representation has many different tumors in it, and these tumors are clustered based on their biological behavior or gene expression profiles. And as you can see, these circles with red color indicating high population consist of gastric tumors, and these blue fraction, or these blue circles, they show absence of gastric tumors. And on the contrary to that, you see that gliomas or glioblastomas, they occupy in this area where there was an absence of gastric tumors. And when we compare this analysis to the actual drug response, we see that tumors in this region and this region were highly sensitive to BYL719, and these two regions are the ones where they were occupied by gastric tumors and absence of gliomas. And this kind of pharmacology drug response was well correlated with when we conducted pathway enrichment analysis, where we compared the PI3 kinase pathway enrichment between gastric tumor and glioblastoma. And as you can see, gastric tumor showed higher enrichment of PI3 kinase pathway, which was very consistent because gastric tumors were highly sensitive to PI3 kinase inhibitor. So all this idea gives us a very credible validation on how pharmacology drug response is well correlated with gene expression profiles. And then the next approach that we did in our study was to able to analyze or compare how each single genomic alteration would dictate drug response to specific inhibitors. And through that, we were able to identify multiple drug interactions, including previously well-established associations, including APC mutation or APC mutated tumors being resistant to a lot of different inhibitors. This was very consistent with previous literatures where APC mutated tumors were highly resistant to a variety of drugs due to the activation of the Wnt signaling pathway. And we also were able to identify KRAS mutant tumors were highly resistant to different PI3 kinase inhibitors, but it was sensitive to both EGFR inhibitor dasatinib and MEK inhibitor. And through that, we have conducted in vitro experimental validation by combining EGFR with the MEK inhibitor, and it showed a very synergistic effect. So this gives an idea how to treat KRAS mutant tumor in the future, that both maybe EGFR combined with MEK inhibitor could provide more therapeutic benefits to these patients. And another interesting association that wasn't really found before was that ibrutinib, which is a BTK inhibitor, was highly associated with EGFR variant 3. So EGFR variant 3 is a structural variation with exon deletion, which is exon 2 to 7 of EGFR is deleted. And because of that, it leads to constitutive activation of EGFR signaling pathway. And this structural variation is well found in glioblastomas. And interestingly, when we conducted a drug cluster analysis, we found that although ibrutinib was targeting BTK, it showed very similar response to other EGFR inhibitors. And as you can see in this graph below, a lot of the tumors were highly sensitive to ibrutinib response, which are clustered on the left side. They had either EGFR copy number amplification or mutation of EGFR variant 3. And then we were able to validate this through in vitro and in vivo analysis, where EGFR amplification and variant 3 harboring tumors show high sensitivity to ibrutinib. And we were able to find that ibrutinib successfully repressed EGFR downstream signaling axis, which consists of STAT3, ERK, AKT, and so forth. And the ibrutinib also was able to significantly prolong the survival of mouse xenograft model. And then through immunohistochemical and immunofluorescence analysis, we found that ibrutinib was able to downregulate EGFR in vivo as well. So this gives us a very good validation of how our gene-drug association could be translated into the preclinical translational research. One thing that we asked, and a lot of people probably have the same question as well, is that is single genomic alteration really enough to be able to predict drug response? And then there should be a lot of variables to account for, but the answer should be no, because drug response is a very complicated process, and the single genomic alteration most likely will not always dictate such a response. So in order to address this challenge, we have used different analytical approach by using integrative multi-layer data analysis, which is the diffusion, or DNetFS, which stands for diffusion kernel-based network module for feature selection. And what it does is it integrates a lot of different layers of data. In this case, we were able to combine protein-to-protein interaction with previous known drug targets that have been found in different literatures, along with our genomic characterization to be able to prioritize which genomic or which transcriptomic expression is most likely to dictate how this drug would respond. So we have applied this approach to a lot of different drugs, and while there was a lot of different interesting associations, we were very interested in panobinostat, which is a HDAC inhibitor. So panobinostat has been rarely been surfaced or used in the field of pharmacogenomics, and there were a lot of different molecular targets that really associated with response to panobinostat. One of them being the high expression of REST was inversely correlated with response to panobinostat. And this is very consistent with previous idea where REST positive tumors, they were very sensitive to panobinostat as well. And while we were able to identify very interesting molecules, what we wanted to pursue after was high expression of HDAC4 and SIK2. And SIK2 happens to be a upregulator of HDAC4, and high expression of these two molecules showed very resistance to panobinostat. And we were able to experimentally validate this using shRNA-mediated knockdown of HDAC4 and SIK2. When these cells were treated with panobinostat, they showed higher sensitizing effect when the cells were infected with silencing of HDAC4, SIK2. And then potential underlying mechanism that we presume is that panobinostat confines the HDAC4 expression in the cytoplasm, and the confined HDAC4 eventually induces activation of oncogenic transcription factors such as HIF-1 alpha and MAP kinase. And this is very interesting because confinement of HDAC4 at the cytoplasm, or the expression of these transcription factors, is also induced by SIK2. So this gives us good validation of consistency between previous literatures and our new findings. So another area that we wanted to explore was there are a lot of limitations on current targeted therapy clinical trials, and especially EGFR alteration or EGFR targeted therapy has really been surfacing a lot, especially in different tumor types, including glioblastoma. So there are different forms of EGFR genomic alterations, including copy number variation, somatic mutation, and structure variation. But although a lot of tumors harbor these genetic alterations, they do not respond to EGFR inhibitors, and we wanted to identify what other factor is causing this, and maybe we can identify new additional biomarker that can be implemented when considering EGFR targeted therapy. So in this study, we specifically focused on glioblastoma samples with EGFR alteration. And then these patients harbored some form of EGFR alterations, including copy number variation, structure variation, and so forth. And then using the similar analytical approach of using DNet-FS and the TDA from previous slides, we were able to identify one molecule that was overlapping between these two analytical approach, which was high expression of NRG1 or neuroglin-1. So high expression of NRG1 was consistently expressed in tumors that were highly resistant to all EGFR inhibitors. And then through experimental validation, we were able to find the silencing of NRG1 or silencing of NRG1 through shRNA or the neutralizing antibody increased the sensitizing effect to the EGFR inhibitors. So this gives us a very good sense of how people should consider performing EGFR targeted therapy because high expression of NRG1 expression could potentially dictate EGFR resistant in EGFR altered patients. So overall, there's a lot of knowledge and a lot of interesting findings here, but the most important step is for the clinicians, how can we translate such knowledge and accumulate data into the clinic? How can clinicians make informed decision based on such analysis? And in order to address this and help very different clinicians at our hospital, we were able to accumulate 31 patients or 31 retrospective cases where the patients were given the targeted therapy and their drug response was available in our system. So we were able to compare our drug screening results with each individual patient who are diagnosed with either gastric cancer, glioblastoma, lung cancer, or atypical meningioma. And when we compare these two factors, what we found to be very interesting was that patients who showed a complete or partial response to that specific therapy were very highly sensitive to that specific drug in our drug screening results. And on the other hand, patients who showed either stable or progressive disease were very highly resistant. And using the ROC curve analysis, it showed that our drug screening results was able to successfully predict the potential response of the patient at a very high confidence interval. And then the example here is basically the pinnacle of what we wanted to convey in this manuscript. What you see here in this graph is two patients who are diagnosed with glioblastoma. Left patient, actually both patients harbored genomic alteration in PTEN. Left patient had a PTEN mutation, while the right patient had a PTEN deletion. And ideally, theoretically, these two patients should react very well to PI3 kinase inhibitor. But in the clinic, this wasn't the case. Left patient, although left patient did respond very well to the PI3 kinase inhibitor treatment, right patient didn't. And then, interestingly, when we conducted drug screening of their patient cells, the results were very consistent. Patient-derived spheroids from the left patient was very highly sensitive, while the right patient didn't. So this gives us a very this gives us very confident results that genomic profiling of the tumor alone is not enough to be able to predict how a patient will react. And in this study, we proposed that drug response of drug screening of these patient derivatives can potentially add or provide additional information that can really make the informed decision to the clinic, because patient can be only treated at maximum, it's very limited. And then we need to be very cautious on how to translate our knowledge into the clinic where the patients are given specific targeted therapy. So all in all, in summary, there are four major conclusions that I'd like to address. The first one being the feasibility and clinical relevance of our drug screening system for pharmacogenomic analysis. And second major point was that therapeutic landscape of these patient-derived spheroids could identify each cancer type specific drug association, including gastric cancers being more highly sensitive to PI3 kinase inhibitors. And we were also able to identify repurposing of ibrutinib for EGFR targeted therapy, which has been an area I think a lot of people are very interested in, because a lot of the FDA approved drugs, they are currently being repurposed or reused into treating cancer patients. And then third major comment that we wanted to convey is that we also are able to identify specific molecular determinant that can really govern how each drug will respond, including HDAC4 and SIK2 to the panavinostat, and NRG1 to EGFR inhibitors. And this gives us an opportunity to explore how combination therapy can be really be implemented. And lastly, we also were able to demonstrate clinical concordance between our drug screening sensitivity to clinical response. And this really presents that systematic evaluation could make a breakthrough points for precision oncology therapy approach. And lastly, at the moment, we're currently enrolling gastric patients so that we can conduct similar analytical approach of genomic profiling along with drug response, so that we can make informed decision for each patient on how they can be treated. Lastly, there are a lot of people that I want to thank in the study. As you can see, our lab is very huge, and there are a lot of people involved in the study as well. This project probably couldn't have been finished without any one of them. And then since there is a very limited time and not enough time for me to thank everyone, I'd like to thank those who are directly involved in this paper. From our group, I would like to thank Professor Do-Yeon Nam and Dr. Jin-Gu Lee, Professor Do-Yeon Nam really supervised the entire project. And our collaborators at Columbia University, Raul, Jauchi, Nicola Antonio. And I would like to give especially thanks to Professor Jian Lee at Hemato-Oncology Department in our hospital. She really provided a lot of in-depth perspective on how clinical aspects should be addressed in this study. And also big thanks to Dr. Sun-Young Kim, who really helped out the revision. And also I would thank collaborators at Jackson Lab, Royal Varrock, and Hong Kong University of Science and Technology, Jigang Wang. And most importantly, I would like to thank everyone who really took the time and liberty to be here today. And also thank the BRIC community for giving me this opportunity to present. So thank you very much, everyone, for joining for today.